Hello, welcome back. In this video, we will cover the differential media that are commonly used in microbiology labs. Differential media are specially designed media that help the microbiologist to differentiate among various types of pathogenic bacteria. It is done with the help of certain chemicals in these media. Different bacteria utilize these chemicals in different ways. This produces visual differences among these bacteria when they are grown. The difference is mostly shown by the change in the color of either the colonies or the media. Other visual clues can be formation of gas bubbles or precipitates. One common agent in differential media is the pH indicator that changes color when the pH of the medium changes. Bacterial growth can change color of media by two means. One is fermentation that produces acid and changes the pH to acidic side. The other is utilization of proteins and production of amines that changes the pH to alkaline side. Depending on the pH indicator used in the media, different colors are produced by the bacteria as shown in this table. The commonly used differential media that we will cover in this video are blood agar, MacConkey Agar, Cysteine Lactose Electrolyte Deficient Agar, Mannitol Salt Agar, Hektoen Enteric Agar, Urea Broth, Sulphur Indole Motility Agar, Sugar Iron Agar, and CHROMagar Orientation Medium. If you are interested in any particular differential medium, you can skip to that particular part in the video. Beginning with the blood agar. The agent of differentiation is red blood cells. After inoculation of blood agar, it is kept for overnight incubation. Difference is created by three types of variation in the hemolysis pattern produced by the bacteria. Beta hemolysis is the complete hemolysis below and around the colonies. Example is Streptococcus pyogenes. Alpha hemolysis is partial hemolysis below and around the colonies with production of green pigment methemoglobin surrounding the colonies. Example is Pneumococcus. And finally, gamma hemolysis. That also means no hemolysis. Example is Enterococcus. Next is MacConkey Agar, that is selective for gram-negative bacteria because of presence of bile salts. The agent used for differentiation is lactose sugar and pH indicator is neutral red. Neutral red is pale yellow at pH of 8.0 and turns to bright pinkish red at a pH of 6.8. The difference is created by lactose fermenting bacteria that produce acid as a byproduct of fermentation and turns the pH of the medium towards acidic side. Color of the colonies and surrounding medium changes to pink red. Example is E. coli. Non-lactose fermenting bacteria produces pale yellow colored colonies. Example is Pseudomonas aeruginosa. CLED is cysteine lactose electrolyte deficient medium that is used for isolating and differentiating urinary pathogens. Cysteine supports the growth of cysteine-dependent coliforms, and being electrolyte deficient medium, it prevents swarming of Proteus species, thus giving discrete colonies of Proteus. The differential agent is lactose. The pH indicator is bromothiamol blue, that is yellow at pH of 6.0 and blue at pH of 7.6. The differentiation is produced by lactose fermentation, leading to acid production and change in the color of medium from blue to yellow. Example is E. coli. The non-lactose fermenting colonies appear blue on CLED. Example is Pseudomonas aeruginosa. Next media is Mannitol Salt Agar. It is selective for Staphylococcus species due to presence of 7.5% salt and is used to differentiate pathogenic from non-pathogenic Staphylococcus species. The differentiating agent is mannitol and pH indicator is phenol red. Phenol red is purple-pink at pH of 8.4 and changes color to yellow at pH of 6.8. Since mannitol is the only sugar in the medium, the difference is produced by mannitol fermenting Staphylococcus by producing acid. The color of the medium changes from red to yellow. Example is Staphylococcus aureus that forms lush pigmented colonies surrounded by a yellow halo due to mannitol fermentation. Non-pathogenic staphylococci form small red colonies without any change in the color of the medium. There are some other Staphylococcus species that are also mannitol fermenters. Example is Staphylococcus capitis, Staphylococcus xylosus, Staphylococcus coheni and simulans. So further biochemical tested may be needed. Moving on to Hektoen Enteric agar, that is selective for gram-negative organisms by inhibiting gram-positive organism with the help of bile salts and dyes in the medium. It is a direct medium for fecal samples to increase the yield of enteric pathogens. It is primarily a screening agar for Salmonella and Shigella species and differentiate them from heavy number of normal flora. It has 3 differentiating agents, lactose, sucrose, salicin, that are all carbohydrates. The pH indicator used is bromothiamyl blue, that is yellow at pH of 6.0 and turns blue at a pH of 7.6. Another indicator is acid fuchsin that turns red in presence of aldehyde. Sodium thiosulfate is a sulfur source for H2S gas that is detected by the iron turning the colonies black in color by the production of iron sulfide. Differentiation is created by the fermentation of lactose, sucrose and salicin by the coliforms and production of acid that changes the color of the medium from blue to yellow. Reddish or salmon colored colonies are shown by commensal coliforms. Pathogenic Salmonella is one of the suspected bacteria in diarrhea or dysentery that do not ferment any of the 3 carbohydrates. So they form translucent light green or greenish blue colonies, while most non-pathogenic coliforms can use at least 1 sugar, so they form orange, yellow or salmon colored colonies. Moreover, Salmonella produces H2S, turning the colonies black in color. Pathogenic Shigella do not ferment any of the 3 carbohydrates, so they form greenish colonies and does not produce H2S. Now we move on to urea broth. It is used for detection of urease producing bacteria. The differentiating agent is urea. The pH indicator used is phenol red that turns from its original yellow color to pink in the alkaline medium. Differentiation is produced by bacteria that hydrolyze urea to ammonia. The ammonia combines with carbon dioxide and water to form ammonium carbonate, which turns the medium alkaline, changing the color of the media and colonies from yellow to pink. Rapid urease positive organisms include Proteus species, Morganella morgani, and some Providentia stuarti strains that turn pink within 1 to 6 hours of incubation. Delayed urease positive reaction is given by organisms like Klebsiella species and Enterobacter species after 6 hours of incubation. It can become more intense during further incubation. Negative urease reaction is shown by persistent yellowish color and is an example of Escherichia coli. Moving on, the next medium is Sulphur Indole Motility, also called SIM. It tests many things at the same time, including sulfur reduction, indole production, and motility in semi-solid agar. SIM media is used to differentiate different members of family Enterobacteriaceae. The differentiation agent in this medium is tryptophan present in casein peptone. The media also has thiosulfate and iron to detect H2S production. Media is inoculated using a straight wire that is first dipped in the bacterial suspension and then a stab is made in the center of the media. Media is then kept in the incubator. Differentiation is produced by the bacteria that degrade tryptophan into indole, which is detected by addition of COVAX reagent after overnight incubation. Indole production is shown by formation of a red band at the top of the medium upon addition of COVAX agent. Non-indole producing organisms do not develop any red band at the top. As the name of the medium suggests, S stands for sulfide, that is H2S production. It is shown by development of black precipitates in the medium. Indole reaction is shown by the appearance of red band on addition of COVAX reagent. M stands for motility. The motile bacteria move away from the stab line in the center of the medium and produces turbidity or cloudiness around the stab line, suggestive of motility. The growth of the non-motile bacteria is restricted along the stab line with a clear surrounding medium. Different bacteria show a combination of these three features, that is sulfur, indole and motility. Let us see some examples. Shigella species have indole positive, but they are non-motile and do not produce H2S. E. coli are motile as well as indole producing, but they do not produce H2S. Klebsiella pneumoniae and Shigella soniae are sulfur, indole and motility negative. Proteus vulgaris is positive for all three, that is sulfur, indole and motility. The motility may not be well appreciated because of the blackening of the media. Salmonella species and Proteus mirabilis do not produce indole but are positive for sulfur reduction and motility. Now triple sugar iron agar or TSI agar. It is a solid medium in a tube with two parts, a butt in the bottom and a slant on the side of the tube. Ideally both should be 3 cm in length. It is used to differentiate members of the family Enterobacteriaceae. The butt provides relatively anaerobic environment and the slant is exposed to air and provides aerobic environment. TSI contains 3 fermentable carbohydrates, glucose, lactose, sucrose that acts as differentiating agents. The pH indicator used is phenol red, that is yellow at pH of 6.8 and pink at a pH of 8.4. TSI is buffered at a pH of 7.4 and production of relatively small quantities of acid result in visible color change. It also has thiosulfate and iron to detect H2S production. Also, the TSI agar is very rich in nutrition by the presence of beef extract, yeast extract, peptone and proteose. It grows most bacterial species, so it should be used for testing bacterial species selected from a single pure colony. Carbohydrates are evenly distributed through the slant and butt of the tube, and the concentration of lactose to sucrose to glucose is 10:10:1. TSI agar should be used mainly for differentiation of members of family Enterobacteriaceae. Before using TSI agar on fermenting bacteria, the cytochrome oxidase test should be performed to differentiate other genera of fermenting bacteria such as Aeromonas, Plesiomonas, Vibrio, Pastorella species that are oxidase positive. It is advisable not to do TSI for oxidase positive fermenting bacteria as it saves time and money. The slant is inoculated with a long straight wire. The process called a stab, and while removing wire from the stab, the slant is streaked with the back and forth motion. Media is incubated after inoculation. We can see 4 types of visible reactions in the TSI agar, that includes yellow color production by the sugar fermenting bacteria, pink color by non-fermenting bacteria, black discoloration mainly in the butt by H2S producing bacteria, and disruption of the media in butt region by the gas producing bacteria. Various combination of these 4 reactions can be seen. Let us see some examples. The non-carbohydrate fermenters that will utilize peptone in the medium and produce amines, making the medium alkaline, indicated by the pink color throughout the medium. Example is Pseudomonas aeruginosa. Some bacteria ferment glucose but do not ferment lactose and sucrose. Glucose is present in limited quantities, so in the first 8 to 12 hours, the glucose is fermented throughout the butt and the slant, producing acid and therefore change in the color of the medium to yellow. In the next few hours, the glucose supply is exhausted and bacteria in the slant switch to the oxidative degradation of amino acids and releases amines, thus changing the pH and color of the slant back from yellow to pink by 18 to 24 hours. The butt, however, remains yellow because there is no oxygen to support oxidative degradation of amino acids in the deep butt. Example of such a bacterium is Shigella species. That is glucose fermenter but not lactose or sucrose fermenter. Some bacteria ferment glucose only, but they also produce H2S in addition. So they will have yellow media at 8 to 12 hours that will turn pink at the slant at 18 to 24 hours, and the butt develops black color by H2S production. Example of such bacteria are Salmonella and Proteus species. The lactose and sucrose fermenting bacteria have sufficient supply of these sugars throughout the medium. The medium is uniformly yellow at 8 to 12 hours. Yersinia enterocolytica is a sucrose fermenter but does not ferment lactose. It has sufficient sucrose throughout the medium, so media is uniformly yellow after 18 to 24 hours. Some bacteria can utilize all the three sugars, that is lactose, sucrose and glucose. These are likely to produce profuse carbon dioxide in the medium and can even disrupt the medium. Example of such bacterium is E. coli. CHROMagar Orientation Medium is a state-of-the-art differential medium for isolation of common urinary pathogens. The differentiating agent in this medium are artificial substances called chromogens. Difference is produced by the release of differently colored compounds upon degradation by specific microbial enzymes produced by certain species. The color-forming reactions are used to distinguish at least 7 species and permits rapid identification and treatment. E. coli shows medium to large size dark rose to pink colonies with or without halos in the surrounding medium. Enterococcus shows blue-green small colonies with halo around them. Klebsiella enterobacter serratia group shows medium blue to dark blue colonies. Staphylococcus aureus shows golden opaque small colonies. Citrobacter species shows metallic blue colonies with red halo. Staphylococcus saprophyticus strains show light pink to rose small opaque colonies with or without halos. Streptococcus agalactiae shows pinpoint to small light blue-green to light blue colonies with or without halos. So that were few commonly used differential media. Hope you liked the video. Please like, comment, subscribe and share. Thank you for watching.